Que le dedicáramos un gran aplauso al doctor Torres Larios. Desde la dirección se está preparando un homenaje que no se había realizado antes por las condiciones de la pandemia, pero que el próximo año se va a hacer un seminario en homenaje del doctor Alfredo Torres Larios. Entonces, por favor, si quieren ponerse de pie y vamos a dedicarle un fuerte aplauso a Alfredo, por favor. Muchas gracias. Bueno, pues es un gusto para mí presentarles al doctor José Arcadio Farías Rico. Él terminó la licenciatura en Biología en la Facultad de Ciencias de nuestra universidad, así también como la maestría en Ciencias Bioquímicas, la cual realizó bajo la supervisión del doctor Lorenzo Segovia en el Instituto de Biotecnología de nuestra universidad. Tiempo después se reubicó en Alemania para llevar a cabo estudios de evolución y diseño de proteínas, adquiriendo experiencia en biología estructural y bioinformática. Durante su doctorado en el Instituto Max Planck, en el campus de Tübingen, bajo la supervisión de la profesora Birte Hocker. Después de su estancia en Alemania, hizo estudios de postdoctorado en los cuales se especializó en el estudio del plegamiento de proteínas en el ribosoma bajo la supervisión del profesor Gunnar von Higne de la Universidad de Estocolmo. Durante su estancia postdoctoral, adquirió experiencia en el estudio de plegamiento de proteínas in vivo, así como en sistemas de expresión libres de células. Actualmente es miembro del Sistema Nacional de Investigadores Nivel 1 y en noviembre del 2019 se incorporó como investigador asociado en el Laboratorio de Biología Sintética del Centro de Ciencias Genómicas de nuestra universidad. Entonces vamos a comenzar con nuestro seminario. Muchas gracias por estar aquí.
Hola, ¿cómo están? Buenas tardes. Estoy muy contento de estar aquí hoy, gracias a la invitación de Francisco. Solo quería decir un par de cositas. Si me quieren interrumpir, perfecto, para que todo quede claro y se haga como un poquito más didáctico, que no sea solo una plática así convencional. Y también estuvimos discutiendo si daba el seminario en inglés o en español, entonces al final decidimos que en inglés, para que algún par de colegas que están, bueno, por todo el mundo, pues también lo puedan ver. Y si hay alguna pregunta, o prefieren no, o y también se puede preguntar en español, o no sé si, o también podemos hacerlo democrático, si a alguien le molesta mucho el inglés, levante la mano y lo doy en español, ¿no? Entonces, pues bueno, voy a empezar. So today we're going to talk about folding and evolution of proteins on the ribosome. So here you can see a ribosome, a nice representation of the ribosome, and a protein which starts to be folded here. And also we have the light from Charles Darwin there. And so we can start from very basic questions like what is a protein, because sometimes some people forget. And so proteins are the expression of the genes. So here we have the human genome, for instance, that transcribed into mRNA, messenger RNA, that is going to be trapped by the ribosome. And then the ribosome is going to translate the protein that many times is represented as a spaghetti. That's when it's just burn the protein, and it's actually not a structure. So in this case, the protein has this primary structure, which is composed by amino acids, and then the protein starts to be folded and reaching a secondary structure that when the protein is making alpha helices and beta sheets, and then these helices and beta sheets collapse into a tertiary structure that is the folded conformation of the protein, which is actually native, the functional, and the process that the protein travels from unfolded to folded is called as protein folding. And so why it's nice to study proteins? Because they are the functional agents of life. So they do every function, everything in the body and in all the biosphere. So here, for example, we can see that they have a very important role in defense against viruses. They also give structure as collagen here, or there are enzymes that there are enzymes as well that, for example, the alpha amylase, which we have in the saliva to start digesting our food, or communication as insulin in this case. So proteins are basically doing everything. And we can also go back in time And we can start asking other more philosophical questions, like, for example, what is the origin of proteins? And if we look back on time, and then we see that the formation of Earth was 4.5 billion years ago, then we see how water appeared a bit later, and then it was likely that there was a prebiotic chemistry here, somehow emulated by the Stanley Miller experiment, where he was able to find aspartic acid, glycine, alanine, among other residue amino acids in the beginning of life, and when life was starting to appear. So these amino acids start like getting together by forming short peptides by wet and dry cycling. And then a bit later, so we start witnessing, or like there is evidences or suggestions that the first protein were high symmetry mini proteins that start associating with a proto-ribosome. This is a hypothesis from Ada Jonath, the one, a very important scientist who won the Nobel Prize for the determination of the structure of the ribosome. And later on, there is the possibility to have real Darwinian evolution when we have genomes that are translated by the ribosome. So this is when everything that is alive translates. So then this triggers the question of whether viruses are alive or not. So, but this is not a question for right now. So then we are going to focus on this part of the graph. So what kind of coevolution patterns we can find about that are together between the proteins and the ribosome. So then this is a representation of the ribosome that is in a way artistic, but it's also very precise. So these are the representations made by David Gossell from the PDB, Protein Data Bank. And this is the factory that makes all the proteins. So we have here the small subunit that is in charge of making the processivity of the translation. And we have also the large subunit, which is the one actually stitching the amino acids together. And we also have the exit tunnel where proteins escape. And we have the proteome. So this is basically the factory and the products of the factory. So you can see that there is a very big structural diversity of proteins that are doing many different things. They associate in complexes. They are sometimes very small. You have heteroligomers, homoligomers, and many different flavors of proteins. We can start with the ribosome. So the ribosome, there are some evidences that it evolves like an onion. So it seems that the most inner layer is the most ancestral. So when you compare ribosomes, you can see that you compare ribosomes from different species. So the more to the center you go, the more similar they are. And the more outside you go, so the more dissimilar they are. And here also I want to like make a scheme or like a representation that at the beginning of time, so the ribosome, the two different subunits evolve independently, and at some point they associated and they start evolving together. So, and here, for example, we have the exit tunnel. This is where proteins escape from the ribosome. So it has a length of 100 Armstrongs. So this is a representation of the exit tunnel that is going to be the star of this talk. One interesting fact about the exit tunnel, especially, so this is just another representation of what I said before. So like one could somehow say that we all have a bacterial ribosome in our eukaryotic ribosome or mammalian, because so the more you go to the center, the more similar they are. And you have a very nice embellishments of RNA embellishments in mammalian ribosomes, for example. We don't know what they are or what they are doing, but this is like an alien embellishment. So it's very different, the two different ribosomes. And we are very lucky because we have structures for a lot of ribosomes at the moment. So there's, with the efforts from cryostructural microscopy, so then we can have access to many different structures of the ribosomes. So this is what these people did. So probably most of you know the three-domain system of classification developed by Carl Guss, where he used the sequence from the 16S and the 18S ribosomal RNA, so the one which is very close to the center. And you can see here how when you have a tree, a phylogenetic tree built by using sequences from these RNAs, so again you can see how the bacteria is clustered together, also with the archaeas and the eukaryotic organisms clustered together. And this, what is very nice from this publication is that the authors managed to derive a metric of the geometry of the ribosome. So they managed to codify the geometry of many different ribosomes from different structures, from different species, and they cluster they cluster the organisms in function of the the distance between the different geometries, how similar they are in geometry. So it's very important to see the difference here. So here we have sequence-based evidence, and here we have geometric evidence, which is not very common to use in evolutionary studies. So here you can see how it's very well reconstructed, the positioning of the leaves of this tree. So you have, again, the bacteria, then you have the archaeas in the center, and then you have the eukaryotic organisms. And when you include the ribosomes from the organelles, like the mitoribosome, for example, or the ribosome from the chloroplast, because they also have their own ribosomes, so they also cluster where they should be, so in the bacteria. So it's actually very nice. This can tell us a little bit about maybe the ribosome is imprinting some signals, some footprints of evolution on the proteins that they are codifying for translating. So if we start talking about protein folding, so we see that for about 50 years or 60 years now, the studies on protein folding have been done in diluted buffered conditions. So when you, as a biochemist or biophysicist, you purify your protein and you have it in a buffer, and then you can follow the folding in a very specific system. So you have only the buffer, salts, temperature, all these kind of things. But now we are trying to because it's obvious that proteins do not fall in isolation. So this is a representation of the E. coli done also by David Gossell, where this is a cross section of bacteria of E. coli. So you here can see the double membrane. You can see the accessory things that are working with the DNA. You can see ribosomes translating, for example, here. And so it's a very complex environment. And what is it important to differentiate between these two scenarios? So many of the studies that have been done in protein folding, so they assume that proteins are completely alone, and they are traveling through this protein folding landscape, where the most energy, the higher energy states of the protein, unfolded ones, are on the top, and then they reach the folded state. So if we see in this animation, so almost all of the protein is available to make interactions with itself. So it can actually, the carboxy terminal part can actually interact very well with the N-terminal part, and then the protein is going to reach folded state. And but this is not the case when the protein is being translated by the ribosome, because this is a time lapse, which is a bit speed up, so we can make it faster. So this is, I think, is 4 times the normal speed. In eukaryotic ribosomes translate slower than the bacterial ones. And you can see here how the protein is going out of the ribosome, but only the N-terminal part of the protein could establish interactions with itself or with other parts of the protein, while the C-terminal part is still sequestered inside of the tunnel. So in a way, you have some kind of a folding box that is allowing the protein to go vectorial out of it. So this is just to keep it in mind, the main difference between in vitro folding versus folding on the ribosome. Now, my lab wants to situate between both. So we try to go not completely in vitro, and we are now moving a bit more to in vivo, but we also we try to be in the middle way of studying protein folding, so with some kind of a middle way of complexity. How we reach this? So we use a system that is was developed around, I think, 5 years ago, a little bit more, by a Japanese group that is called the Pure system. So this actually is one of the main achievements of synthetic biology, modern synthetic biology, because we have a very deep understanding of translation at the moment. So we can actually purify every single element that is needed to perform translation in bacteria. So in this case, for example, the ribosomes are purified, every single amino acylternase synthetase, all the amino acids, energy, translation factors, and if it's coupled system, so you also have a T7 RNA polymerase, and this system is a highly purified system, is called the Pure system, and you can get it in Mexico from New England Biolabs, but it's also super expensive. So, and it's actually a bit prohibitive to work with this in Mexico at the moment, and but then once you have it, you can start learning a lot of things about protein folding. So for this, I'm going to talk about the method that I will be discussing during the talk. So this is really important that everybody understands. So if I don't explain it properly, you can tell me and I repeat it again, maybe even in Spanish. So it's using the Pure system. What we do is, if we want to study a protein, we want to understand the folding of a protein on the ribosome. So we create this genetic construct. So this is a construct that is in a plasmid. And then we have our protein of interest. Then we have a flexible linker with a variable L. And then we have an arrest peptide. And this arrest peptide is the start of the method. And we have a carboxy terminal extension. So this is a cryo-electron microscopy of an arrest peptide. So this is in green. And these are the walls of the ribosome. So here you can see the zoom out figure of the ribosomes. You have the small subunit, the big subunit, you have the tRNA still stuck there. And what happens when the ribosome is translating this translational fusion is when it reaches the last proline of the arrest peptide, so it cannot continue further. It gets stuck there. So this arrest peptide has a biological function that is not necessary to discuss today, but what is it doing is it stops translation. So the ribosome actually gets stuck there. And it cannot continue translating. So in the case, and then using our method, what we will have, so we take this sample, we go to the radioactive room in the CCG, and we put our label in methionine to be able to see our proteins, and we start translation. So the ribosome is going to start translating, and it's going to reach the last proline, and it's going to sit there. If the linker L is too short, so the protein doesn't have enough space to fold, and there is no release of chemical energy that is not translated as mechanical force. If in a second situation, when the linker is long enough, what will happen is that you have enough space for the protein to fold, then the chemical energy, the free energy of Gibbs that the protein releases during folding is going to be translated as a mechanical tension, as a mechanical force. So it creates tension on the chain, and it releases the arrest. And then in this case, so the ribosome will be able to translate the whole construction, the whole construct. When there is no space, the ribosome translates up to the arrested version. And then when the protein is too far away from the ribosome, there is no influence of the ribosome anymore. So the protein folds, and but there is no release of the arrest because then it doesn't have anything against to push. So the protein cannot really push because it's too far away from the ribosome. Imagine that you have something hanging around, but it's not really making interactions with the ribosome. So then what we're going to have our readout is actually really simple because we are going to have two gels in a band, two bands in a gel, sorry. So in this case, again, just to remember that this is the pure system, and this is a exemplary of our constructs. I'm not wanting you to read the sequence, but so you have the protein here, and you have the linker that is increasing in size. This is what the ribosome can accommodate inside of the tunnel, what is highlighted in red. And then you have the arrest peptide and then the extension. So what is going to happen if we take one of them, for example, let's say that we take the L40 here. So we run this, we express it for 15 minutes, 37 degrees, and then we run it in an SDS page, and we have controls. So the controls are done to identify the bands in the gel, and then we have a band that represents the full-length protein, and we have a band that represents the arrested one. So we have a ratio between these two bands. So this, in a way, represents the population in the test tube of the proteins that were able to fold and the proteins that were not able to fold. So in a way, if you think about kind of molecular population or population molecular, so then you have population of molecules here. And then we get a ratio of these two. When we quantify this is very much quantitative, so you can get a ratio, and then you get a fraction full length. So it's basically the intensity of the full length divided by the full length plus the arrested. So it's a very simple formula. Then we do this for every single one of the constructs, and then we get a profile, is the one that we show here. So the fraction full length in function of the L, of the linker length. So this basically tells us how close to the center of the ribosome the protein is folding. So in this case, for this specific protein, the protein is the maximum folding force that is released by the protein is around 40 residues away, 45 residues away from the center of the ribosome. So this is actually tell us, because just to remind you, you can accommodate about 35, 40 residues in extended conformation in the tunnel because it's 100 angstroms in length. So I hope that this is clear. If not, then I just say it again. And because we did a lot of work with this, and one of the most difficult parts was to convince the reviewers that we were actually measuring folding, because classical biophysicists do not, I mean, sometimes it's for them hard to believe that we are actually measuring folding. So what we did, we work in a collaboration with Professor Mikael Oliver in Stockholm University. So what he did with this protein is that it's a model protein that is being used quite a lot in classical biophysical folding studies. So he mutated 18 residues of the protein to alanine. So, and then in this case, he could estimate very carefully every single mutant to measure the thermodynamic stability in vitro. So he made unfolding curves for every single mutant. So it was a lot of work, a lot of mutants, and we get the constants, the true thermodynamic constants for every single mutant of this protein. So then we took that one and we measured it in our system, and we actually make the mutations as well. And we plot the fraction full length in function of the thermodynamic stability loss of every one of the mutants. And then you can see that there is a very nice correlation that is being accepted now a little bit by the community, that we are actually measuring thermodynamic stability. And now we have more evidences from all different groups that now adopted the method, and now we can be sure that we are measuring thermodynamic stability. So with this method, we did a tour de force in a way. So we measured the folding of several different proteins. It was the work done by these amazing students I had in Sweden, by Joanna and Frida in Stockholm University still. So we measured the folding of the bigger proteins to very small ones. So we wanted to estimate how the size of the protein affects its folding on the ribosome. So we wanted to see how it will work. No? So we don't have the time to go with every specific detail of this, but if we sum up the data, so we see that everything which is below this blue line is folding inside of the ribosome, and which is folding outside, which is above the blue line is folding outside of the ribosome. So this actually has very interesting and functional implications, because a lot of post-translational processing will, I mean, for example, in the case of bigger proteins, well, it has to happen outside. But what happens with the small proteins that are folding inside? So they are not targeted by chaperones, for example, or like this actually prompts a lot of questions. How is the processing after the protein is being translated?
So you're saying that inside the tunnel exit, you can have a folded protein?
Yes.
Is that wide enough?
Yes. Yes. For a very small ones, for example, or when it depends how they accommodate inside of the tunnel, they can also be just a little bit stuck there. And there is actually cryo-electron microscopy pictures of Titan, for example, which is very well folded, just right in the mouth. And for example, in the case of this one, which is ADR1, it's a zinc finger domain that is super small. Some people will say it is a peptide because its stereo ratio is long, but this has very much space and very deep of the tunnel. And there is a cryo-electron microscopy as well from this one. So.
What's the purpose of being able to make structural changes to the protein while it's being folded? Because bigger proteins are less stable?
Well, I mean, many proteins need post-translational modifications to work, and this is what we are actually trying to discover at the moment. So we are trying to understand if these processes happen post-translationally or co-translationally, while they are still being translated, and we are trying still to measure the impact that this new knowledge is giving us. Like what's going to happen with the post-translational or co-translational modification of these small ones. So what happens when you add a phosphate or when you add another special thing to the protein, and how it happens. So this is something that we still do not know very well. Yeah.
Have you tried the membrane protein?
Yes, actually this method was very helpful and was discovered by the Gunnar lab. Gunnar von Heine, probably you know about him. So he is very big in membrane proteins. And this is how he discovered the method. So he developed it because when membrane proteins are targeted to the membrane, the ribosome is completely targeted to the membrane, it goes through the translocon. Then when the protein is going through the translocon, then it is when it's experienced the pulling force. That's why this is how it was discovered actually with membranes. And there is a very nice new publication from Gunnar's lab and from Felix that was doing his PhD when I was there. And I can give it to you later if you want. So it just appeared last month. It's actually very, very useful for membrane proteins. Yeah. And another finding that we got from this work is that also the charge, it's not only the size, but also the charge is very important for, it's going to have a strong impact on the folding of the proteins. For example, so how it works in this case is that we have this mutant S6 protein, the same one that we used before, and this one is completely devoid of charges, which is also very interesting, no? Because it's actually a protein that can fold without any charge. So it's like very weird. And so we use this one, and we measure the folding on the ribosome again, and then we see that it has some kind of a very nicely shaped force profile that is folding around 35 to 40. But then when we measure the same protein and it has a net charge of -17, so this protein is very highly charged. So then, because you remember that what we are measuring is tension in the chain, so then we have just a completely different profile, and it seems that the protein is just being repelled by the surface of the ribosome. Because here we see the space filling representation of the 4 proteins that are making the exit tunnel is UL4, UL23, 24, and 22. And here you can see that there is a very differential charge distribution. And also, if you remember, the ribosome is made from RNA, and the phosphates have a very strong, so in total it makes a very strong negative surface. So then this is why this protein is being repelled quite a lot. And we actually measured a supercharged protein on the positive side, but we were not able to measure it because it got stuck. It actually didn't translate. And this gives us hints about how it could actually impact the proteomes, because if you see here, I mean, it's not so easy to see, but there are different proteomes they are shown in the net charge. And so probably the ribosome is having an impact, a differential impact in the net charge of the proteins. So it will have interesting evolutionary implications. And with this, I can close the first part of the talk, making a little summary where the ribosome now is being seen as some kind of up the hill energy hill. So it helps proteins to reach different energetic states that wouldn't reach otherwise. And for example, proteins that are very large and they fold through kinetic control wouldn't be able to fold without the ribosome. And now this very classical protein folding landscapes that we always see that are only in some kind of a flat surface, well, now they start so much higher. And so in there are some of the assumptions that we get at the moment, and we think about the ribosome as probably the ultimate chaperone. Yes?
So one comment, one question. The question, if you prefer, you can send it by the end.
Yeah.
The question is, I mean, you're saying that there is definitely it's a different folding, the one that happens in the ribosome versus in vitro. Yes, the structure, the final structure is the same.
Okay. I mean, yeah, we
So
not.
you can discuss this later.
Yeah. But actually, I have an example to show that is not always the case.
Yeah, there are some exceptions, but the general rule is that in general the folding holds, right?
Yes, but also, like, for example, the bias on the proteins that have been determined that the structure is really much biased to proteins that are very small, or like no more than 200 amino acids. And you see the distribution of the proteins in the proteomes, you have hundreds of thousands of proteins that are 500, 300 residues that are not able to get structures. So actually this is also one of the main points, like, so these proteins, probably they will need the assistance of something else, no? Like, I think a third of the coli proteome is not intrinsically refoldable. So I think it's, in this case, yeah. But I have an example. Yeah.
The other is a comment. We've been studying the packing, the density of packing in protein structure. And we noticed that there is a hint in the distribution of this density. So if you will plot the length of the protein versus the packing, it seems to be a linear relationship. Yet, there is a kink. For shorter proteins, there is a higher density of packing than for longer ones. I don't remember right now the length, but it would be quite interesting to see if that matches what you have measured.
Yeah, absolutely.
Because I haven't found any explanation why for small proteins you can get higher density than the larger ones.
Okay, cool.
But the two of them will follow the same packing rule that was proposed by Kepler 400 years ago.
Okay. Yeah. I mean, if you want, we can. We are going for lunch later. Okay. You can. And so then we go for the next part of the talk, and then we will talk about a little bit more about the proteins now. In this case, I mean, this is a bit outdated, so there are more structures now. And then if we include here all the structures predicted by AlphaFold, then it was orders of magnitude higher. And it's still the question whether those structures are very precise or like true. and you have some protein folds, no? So, and then how we can, then we started with the question, how can we make sense of all this structural information and sequence-based information? So for this, we can, I mean, this is very basic, but this is just like, just to hammer the point that we can use evolution to start making like families and groups of proteins that are related in evolution, no? So here we have, for example, an example of proteins that are the antifreeze proteins that are a very nice example of convergent evolution, when you have the same structure converging to the same solution because of, with the same function, no? So they bind the surface of ice crystals with the threonine. So then these are most likely not coming from the same common ancestor. And on the other hand, we have the myoglobin that is very likely that it's coming from the same common ancestor. So in this case, we will be talking about homologous proteins. In this case, we are talking about analogous proteins. So. And so during my PhD, so where I wanted to answer this question, if proteins from different folds were actually homologous, or you could find evidences of homology between different folds. So this is coming from the scope classification of proteins, where you have proteins classified by class, fold, superfamily, and family. So these groups here are believed to be homologous. And then the question was if we could find an homology evidence of this. And the only way that you can have homology completely bulletproof or almost is if you compare sequences, because it's very unlikely that two sequences converge to the same, two proteins converge to the same sequence. It's super unlikely, statistically speaking. They sometimes can converge to the same structure, but it's not the case with sequence. So what I did during my PhD when I was still at the Max Planck, so I compare all the proteins that were classified as T-Barrel fold, clustered in superfamilies, and I did the same with the flavodoxin-like fold. And then we made this hidden Markov model comparison. So we aligned hidden Markov models. So this is sequence-based information that is extremely sensitive. So, and then we found a connection, a sequence-based connection between two superfamilies of this fold that are hitting several families of the other fold that seems to be monophyletic. It's likely. So, and we went a bit further. We continued digging a bit more, and we found an intermediate family of proteins that was linking in sequence both folds. I determined the X-ray crystal structure of this one. And then we discovered that it was a part of the three proteins that connected all of them. So it was very similar in sequence and similar in structure. And then we found this kind of evolutionary intermediate. So it seems that it starts to reveal itself that nature uses these evolutionary successful building blocks to build proteins. So they use the same building blocks in several structural contexts to create all the structural diversity we see today. So this is a database of these building blocks. It's called Fossil. It's accessible from right now, and it has many more additions and things that happen now. And this prompted the question. And one day I was reading the literature, and then I found out that well, this is one of the main questions that we want to answer is it the ribosomal tunnel acting as an evolutionary force, which is impacting the structure of the proteome? So if you see on this part of the slide, so this is a very nice work from the lab from Andrei Lupas in the Max Planck as well. And what they define a vocabulary of ancient peptides at the origin of folded proteins. So they use sequence and structure level information to get a vocabulary of peptides. There is, I think, it's 40 peptides that are very prominent in all different protein folds. And one of them is this zinc finger domain that is this is called here. This is important in regulation of transcription, and it binds zinc. And many of these peptides are involved in binding RNA. So it seems that there is even more strong support for the RNA world hypothesis. And it's the same protein that it was your question whether you could find a protein folding inside of the ribosome. Well, this is the one that was the first one that was determined with the cryo-electron microscopy. And it was published in the Gunnar's lab. So it's basically the same one. No? And then, in this case, this is triggered the question of how we can explore this. Now, this is a work that we've done here in the CSG now. So we wanted to continue exploring this question. And for this, we use a repetitive protein, the Ankyrin repeat protein. This protein is used for many different purposes as well. So to develop DARPins, for example, and the Andreas Pluktung. And so you can see that the protein is built by repeats of the same motif. And these repeats are very likely related in sequence. So you can see that the sequence is very similar. So it's very likely that this protein evolved by duplication and fusion of the same repeat several times. So then we took this protein And we, again, use hidden macro models to search the protein sequence database with the protein. And we hit 3 different folds with different probabilities. And we discover one of the repeats, especially the second one, was hitting the 3 different folds. So in a way, we detected this repetitive share fragment among all these different folds. And then this was a very nice example of so we thought, okay, this protein seems to be folding stepwise, like how it's going out from the ribosome. So we could probably follow it in the ribosome because it's like this stepwise folding. And this is what we did. So this is one of our force profiles. And this is a very long one, because this is the peak of the protein folding completely outside of the ribosome. And this is how it looks like. So for example, we see here that we have a very strong signal for the full folding protein, and the one which is arrested is way smaller. Then we make a mutation that actually brings the thermodynamic stability of the folded protein to almost 0. And then also the fraction full length goes to the kind of noise level. And then we also detected another 2 peaks that correspond to versions of the protein that are made at 1, 2, 3, 4, 5, and 1, 2, 3, 4 repeats. But very interestingly, this is still puzzling us, is that if we make the same mutation in these so-called intermediates, we don't see that the same loss in fraction full length. So in a way, maybe, I mean, one could argue that the protein is folding through a different pathway, or is establishing different interactions when it's some kind of intermediate. We don't know yet. But this, in a way, goes really well with the studies, biophysical studies done many years ago by the Barrick lab, where he actually characterized very truly this protein. And he sees that a protein that has only 4 repeats is just marginally stable, so it's close to 0, the delta G. And when you start adding repeats, it goes to -7. So then you see that the repeats are actually, the interface between the repeats is increasing the stability of the protein. So this was, you can see how this relationship is established there. And now this got recently published, and it was mainly the work from Soledad and Alberto in my lab. And in this case, so we have this little model where the ribosome could have an influence in regulating the folding. So we have a protein that is very, very stable, has 7 repeats, then has 5, 4, and then 1 repeat, that we didn't really see like a real folding peak, but it's kind of dynamic. So maybe there is some kind of something going on there. So we are going, we continue following up here. We trying to design folding if we can design, for example, a metal binding site. And now the rest of the projects are current projects. And this is the example I was talking before. So this is coming from a nice comment on the work from Patricia Clark. And what this comment is just expanding the Amundsen principle, no? Because we all know that all the information for a protein to be folded is codified in its amino acid sequence, but maybe there is even more information there at the DNA sequence. So how the experiment works is that, so when you take a protein that is able to give a fluorescent signal in equilibrium, so this protein can have either signal with the yellow or blue. And then when you refold the protein in vitro and you let it to equilibrate, so then you have like a 50-50 signal. Then if you fold the protein coming out of the ribosome, so because the N-terminal part of the protein is able to establish interactions first, you see that the equilibrium is a bit skewed to the yellow. So the conformation of the protein is more skewed to the yellow part. Then what Patricia did is she engineered actually rare codons, and then she made the ribosome to pause even more. So it made a longer pause, and then the actual equilibrium is almost completely turned to be yellow. So in this case, so the ribosome is actually picking the structure, no? Even if it's the same sequence, no? Can have 2 different conformations. And also this work was follow up, or like it just the line of thought continued by Eugene Sagnovich in Harvard, where he developed this algorithm to detect evolutionary selection for correlational folding. What he did, so he actually measured the frequency of rare codons enrichment on genomes, and then he correlates this with a coarse-grained model for simulating protein folding on the ribosome. And when you see that there is an enrichment of rare codons, then you can see that there is a very strong drop in energy that is indicative of a folding intermediate around 30 residues away, because you remember that the tunnel can accommodate around 30 residues. No? So then you can have intermediates, probably like some kind of intermediates folding outside of the ribosome. So the ribosome sits and waits until this part of the ribosome folds, and then it continues translating. So then we implemented this bioinformatic pipeline in my lab, and this is mainly the work started by Alex. It's continued by Mitch, and now it will probably be finished, hopefully, by Enrique. And so here we have an example of the reads we get. So we have the estimation of rare codons here. When this line goes below 0, you have rare codons, and when this line drops massively, like in this case, well, you have a predicted intermediate on the N-terminal part, and this somehow correlates with the halves of the barrel. So like probably half barrel is folding, and then the next half. And this is still very preliminary, and we don't have experimental evidence for this yet. The experimental evidence that we have so far is the work from Yanni, which she's comparing how proteins fold in the pure system with the in vivo condition. No? So this is almost one of the last experiments. So in this case, what she's measuring again is the folding of the protein in the pure. So you see a folding peak of GFP in this case. So this is like the traditional one that I've been showing you during the talk, and you have a folding peak at around 29, 30 residues away from the center of the ribosome. Then she does the same in vivo. So this is in E. coli and BL21 with this condition of expression conditions. So we actually follow fluorescence here, and then you see that it's kind of the same pattern. So these measurements are still very noisy and very preliminary, but it seems that we are actually reconstructing what we can see in the conditions of the pure in vivo. And why we want to do this? Because we would like to develop a biological processes reporters in vivo. So how this will work? So this is a nice image I took borrowed from one paper from Ed O'Brien that is called Mechanochemistry in Translation. So what happens here is that when a protein is folding, so it generates a force on the exit of the ribosome, and then this force is transmitted to the center, and the force is applied in the PTC, in the ribosomal PTC in the center. So many different cellular processes will actually produce this force, like it will be involved in this one. For example, translocation or membrane insertion, binding, dimerization, and even maybe catalysis, but we don't know yet. And so the idea is to start developing this kind of reporters. And how we do this, so this is already done by Alberto, a very talented student in my lab. So what he did, he actually cloned GFP in this part of the construct after the rest peptide, and then he is been measuring this the folding of this beta propeller, a design beta propeller that we have in a collaboration, and he measured it in vitro with the ribosome and the pure system, and also in vivo. And then you can see that because this protein is folding at around 40 residues away from the ribosome, well, the protein is folding, it releases the arrest, and then you can see expression of EFP. So this is like the first steps to develop a folding reporter in vivo, where you can imagine that you could screen variants of proteins in high throughput manner with many different variants. If you put your protein here, you have to find the sweet spot where the protein will fold, and at this spot, you could probably try to measure whether you could see increases in thermodynamic stability, that if we manage to get really fine in these kind of measurements, we could probably reach it. And the last project that we have, which is probably the most important one, is the one done by Lady, Alex, and Emiliano, he just joined the lab as a master student. And in this case, what we want to do is to produce our own homemade pure system, because as I mentioned before, it's very expensive. So in this case, we were very lucky because this Swiss group published a method how to produce it a couple of years ago, and this is the control. No, so these are the ribosomes that the New England Biolabs provide, and then this is the ones that they actually produce, and these are ours. So this is the first batch of ribosome production in our lab. So you use a sucrose cushion. So you put your lysate here, and then you centrifuge overnight in an ultra centrifuge, and then you can have a pellet of ribosomes. And now we move forward and start working to purify the rest of the proteins, and hopefully we can have a fourfold cost improvement when this is ready, because then we can start establishing more collaborations. Yes?
¿Por qué necesitas un sistema tan puro? ¿Por qué no reticulocito? Si tienes que purificar al final, ¿no?
Sí, pero en el caso, por ejemplo, de si queremos medir interacciones con chaperonas, cuando ya tenemos el lisado, no sabemos si el lisado trae chaperonas o no activas, por ejemplo, en este caso. Sí. And with this, I would like to finish. So thanking to you for your attention and also to the people in my lab. So we have Emiliano, Lady, Sol, Yanni, Alex, Enrique, Mitch, and Alberto, and also to Gunnar. He was really supportive in my move from Sweden to Mexico, and Berlin, who was my last European student. So this is our building in the Center for Genome Sciences. It's the new building in the end of the center. So here are mainly scientists like around my age. So it's like kind of a nice environment to work with. So it's like we are 9 here. And then when I'm in the lab, we like to go to Las Estacas because it's very close by too. It's very nice. And of course to the funding agencies, because doing synthetic biology is really expensive. So without the funding from CONACYT, the GAPA, and Stanford, we couldn't actually do the work. And we are at the moment recruiting students, postdocs, or collaborations are warmly welcome. So thank you for having me today. Yeah. Gracias.
Dado, si bien el concepto de que se plegaban dentro del túnel es para mí totalmente, pues, me movió el piso.
Sí.
¿Sí? ¿Se puede plegar una proteína dentro del túnel?
Para la transmisión de...
Ya. No, sí estaba prendido, ¿no? Creo. Sí, ahora sí.
¿Se puede llegar a plegar una proteína y recobrar su actividad biológica dentro del túnel? Eso te lo pregunto porque se me estaba ocurriendo que si tú tienes una secuencia expuesta en el túnel que fuera reconocida como si fuera un ligando, y tú pliegas una proteína en el túnel que fuera como un receptor, quizá a lo mejor la interacción sería lo suficientemente fuerte y ocurrir lo suficientemente rápido como para que hubiera esa interacción y se atascara ahí la proteína por una interacción específica ligando receptor. O ¿qué más? Una quinasa que se pudiera ligar dentro del túnel, y si tiene disponibilidad de sustratos que fosforilara algo dentro del túnel. Es decir, la actividad biológica de las proteínas dentro del túnel se podría detectar y podría servir de algo para estudiar estos fenómenos.
Sí, es una pregunta interesante porque de hecho apenas empezamos a descubrir lo que podemos medir, porque bueno, eso definitivamente nos va a permitir medir cosas, ¿no? Pero por ejemplo, hablando específicamente de una función, ¿no? En este caso, esta proteína que es un zinc finger domain, pues su función es unir zinc, pero bueno, también es un dominio activador transcripcional. Pero bueno, esta proteína es parte de un dominio, claro.
Eso nada más suelte todo y los créditos, no también va como para unir a alguna secuencia de proteínas.
Pues no, esta no. O sea, sí, esa es nomás la parte del puro zinc, de binding of zinc. Pero, por ejemplo, en este caso, la proteína une zinc adentro, ¿no? O sea, para poder tener esta señal de plegamiento, tuvo que estar adentro. Y estos son 25 residuos, está bien adentro del ribosoma todavía, y ahí es cuando está uniendo zinc, porque si le ponemos tipen a la reacción, pues no hay ningún perfil de plegamiento, ¿no? Es para secuestrar el zinc. Entonces, es un quelador de zinc. Y por ejemplo, en este caso, hablando específicamente de cosas que, a qué nivel podemos medir cosas, este fue el trabajo de Sara, otra estudiante que tuve en Estocolmo, en el cual estamos viendo, lo que ella hizo fue un alanine scan y un tryptophan scan de toda la proteína. Entonces, aquí podemos ver, por ejemplo, que cuando cuando ella mutó las cisteínas y las cistidinas por alaninas, pues la fraction full length bajó bastante, ¿no? Y en el caso, por ejemplo, también de la fenilalanina 12, si se cambia por alanina, pues también hay un efecto en la estabilidad de la proteína bastante fuerte, aunque no es de los residuos que están uniendo el zinc, pero en el caso de cuando ponen triptófano no pasa nada, ¿no? Porque es pues casi el mismo aminoácido y no se destruye el core hidrofóbico de la proteína, ¿no? Entonces, pues al momento estamos descubriendo todo lo que se puede hacer. O sea, también se nos ocurrió la idea de un biosensor, por ejemplo, con esas proteínas que unen, por ejemplo, gliofosfato, y que es como un flytrap, Venus flytrap, que la proteína hace así, cuando lo une, si está bien en el sweet spot, o sea, lo que hay que encontrar primero es el sweet spot de folding, ¿no? Entonces, ya que la encontramos ahí, entonces ahora sí podemos tirarle, por ejemplo, cleofosfato y ver si hay un cambio conformacional fuerte que lo podamos detectar. Y entonces ahí podemos ver fluorescencia o podemos tratar de tener hasta, no sé, en un momento muy lejano, constantes de disociación o algo así, ¿no? Con el ribosoma. Pero eso es como un sueño todavía.
¿Cómo funciona?
El péptido de arresto.
El péptido de arresto.
Este tiene una función biológica que a veces se me olvida, pero básicamente es parte de este sistema de SEC-YEG, en el cual la proteína, el péptido de arresto, o sea, el ribosoma va traduciendo y cuando llega el péptido de arresto hay un lugar aquí de arresto, ¿no? O sea, aquí se para porque está y se tiene que parar para que el ribosoma como que deshaga este hairpin y pueda seguir traduciendo CK, ¿no? Entonces, como que el ribosoma se tiene que detener aquí para unwind the thing, ¿no? Entonces, esto se va a dar porque CKM es incorporated into the membrane, ¿no? Entonces, funciona como para, funciona como para regular el export de CKM, bueno, dice CK, aquí de todo este sistema. ¿Con qué? Un atenuador. Ese no lo conozco.
Hay que leer la historia de la biología molecular antigua.
Sí. Sí. Sí.
Este es otro comentario. Mencionaste brevemente este interés que tienes de ver el efecto que tiene los codones raros en la cinética de plegamiento. Aquí, en el instituto, hay una historia muy bonita que yo creo que puede ser interesante para tu sistema de estudio. El doctor Gómez Puyú hace muchos años publicó un trabajo que publicó en PLOS ONE, en donde él mostraba cómo una variante que él no diseñó, sino que le apareció sin que él quisiera, que cambiaba un codón de arginina por uno de bajo uso en E. coli. Era la triosa fosfato isomerasa de humano expresada en E. coli. Esa variante tenía propiedades cinéticas de plegado y de actividad diferentes a las que tenían el codón silvestre, el codón de alto uso.
Ese sería un excelente ejemplo para utilizarlo en el laboratorio.
Yo creo que ese te podría, porque esa proteína ya está caracterizada.
Ya se sabe,
de hecho resolvieron la estructura.
Pues muy bien, hasta lo voy a anotar, así tener eso. Pero como, bueno, ahorita mejor me lo...
Órale.
¿Quién lo publica?
El doctor Gómez Puyú, Armando Gómez Puyú. PLOS ONE, no me acuerdo del año, pero debe haber sido antes del 2010.
Suena muy bien. Sí, porque en este caso es importante tener
ese control.
Sí, efectivamente, ¿no? Como cosas que podamos, porque lo que queremos es detectar esto como de manera high throughput, ¿no? Pero también queremos, luego a veces es como mucho gigo, ¿no? Como que no, todo lo que predices no es cierto, pero sí, con esos ejemplos, pues puede ser que sí, o sea, estaría padre tenerlo. Gracias. ¿Otra pregunta? Ya.
Bueno, pues, muchas gracias a todos.
Gracias.